All right, let's look at autosomal inheritance patterns for genetic disorders and pedigrees and how you do those. So pedigrees are basically just tracking the genotypes of different generations. So you've got a parental generation or a P1 generation, the first level of offspring or F1, and then the second level of offspring or F2. I'm doing the second pedigree as the example because it's a little bit more complex than the previous pedigree in your lab manual, which has to do with earlobe attachment. So if you can do this one, then you should be able to do the earlobe attachment no problem. In an autosomal recessive genetic disorder, the important thing to remember is that the individuals with the homozygous dominant genotype are free of the disease. They are completely normal, they have no problems. If they have the homozygous recessive genotype, then they have the disease. And if they have the heterozygous genotype, they don't have the disease, but they are considered carriers of the disease because they could potentially pass on that recessive allele to their offspring, and then their offspring could have the disease if they got two recessive alleles from both parents. The particular disorder that we're looking at today is sickle cell anemia, which is an autosomal recessive disorder, which means that if you have a dominant allele, then at least you don't have the true disorder. If you have two recessive alleles, though, then you've got it. In your lab manual, it's giving you this pedigree, and they're giving you some of the genotypes, but not all of them. So you have to figure out what the other genotypes would be based off the information they give you. So let's look at this. So this first thing, it says two of the parents of the F1 generation. So the parents of the F1 generation would be the P1 generation. Have genotypes that are shown in the figure. Predict and fill in the answers for the other parents' possible genotypes. What are the potential genotypes of the F2 generation? Well, let's look at the P1 generation first. So for this F1 generation, we have the squares are male and the ovals are female, so we have a husband and a wife, and they are both carriers for the disease. They're both heterozygous individuals, which means that that dominant allele, the uppercase N, had to come from one parent, and the recessive allele had to come from the other parent. Looking at the P1 generation, we can tell that the father, because it's a square, so that's male, the father for the P1 generation has two recessive alleles, which means that he donated one of his recessive alleles down to his son, and that's where that son got the recessive allele. That means the mother must have contributed the dominant allele, or that uppercase N. So we know that one of her alleles has to be a dominant allele, but we don't really know what the second one is that she has, and it could be either one. So they're offering you, there are two different options. So the maternal grandmother could have two dominant alleles and be homozygous dominant, or she could have one dominant allele and one recessive allele and be heterozygous. But either way, according to this pedigree, she donated that dominant allele to her son. Let's look at the woman, the female of the F1 generation, and look at her parents. So the female of the F1 generation is heterozygous as well. She has a dominant allele and a recessive allele. The father of that female has two dominant alleles, two uppercase N's. So we know that one of those dominant alleles was donated down to her, and that's why she's got a dominant allele here. But she's also got a second allele that's recessive, which means that her mother must have donated that recessive allele. But once again, we only know that the grandmother up here had a recessive allele. We don't know what her other allele was. So there are two options. She could have had two recessive alleles, or she could have had a recessive allele that she donated to her daughter, and a dominant allele. So this is the P1 generation, the paternal grandmother and maternal grandmother. When you're comparing them to the F2 generation, these would be the grandparents. So that's why they're saying that, paternal grandmother, maternal grandmother. All right, now let's look at the children. The children would be the F2 generation. So if the parents, which are the F1 generation, are heterozygous, then we know that there are four possible outcomes or four possible variations for the genotypes of the children. And we know that, just like when we were doing previously, because with the normal Punnett square where you're crossing two heterozygous individuals, you got these four options. Two of them are the same, because two of them are heterozygous. One is homozygous dominant, one is homozygous recessive. So, looking at our lab manual, we have four options. We can have a child that is homozygous dominant. We have two options for heterozygous. and then we have a homozygous recessive. And those are our 4 options for the children. Okay. The next question's a little bit weird. They're basically wanting to make sure that you understand the relationship amongst all the different levels of the pedigree. So the question says, what are the possible parental genotypes of the F1 generation? Well, the parental, or the parents of the F1 generation, which is this level right here, would be the P1 generation, which is this level up here. So I put that in red. That's basically asking about the possible parental genotypes of the P1 generation, and then what are their phenotypes? Well, the genotypes have actually already listed over here, and they're listed on the pedigree. So the paternal father would be this square up here. His genotype is homozygous recessive. So his phenotype is that he has sickle cell anemia. The paternal mother would be this oval up here. We already said that there are 2 options for her. She could either be homozygous dominant, or she could be heterozygous. If she's homozygous dominant, then she would be considered normal. If she's heterozygous, then she'd be considered a carrier. The maternal father would be this square up here. We are given the information that he is homozygous dominant, and that would mean his phenotype is that he is normal. The maternal mother would be this oval. We already determined that she has 2 options, but she's got to have 1 recessive allele in either option. So she could either be homozygous recessive or heterozygous. If she is homozygous recessive, then she is going to be affected. So she would be sickle cell anemia affected. If she is heterozygous, she would be considered a carrier. All right, so the next question says, what percentage of the F1 generation individuals are normal, and which ones are carriers? F1 generation, technically both of these are carriers, so we would say 100% are carriers. Oops, not 200%, just 100%. And 0% will be considered normal. Next question, what percentage of the F2 individuals are normal, are carriers, or are affected? Let's look at the F2 generation. That's the children. So looking at the children over here, we can say that 1 child, or 25%, would be considered normal. 2 out of 4 would be considered carriers, that'd be 50%. And then 1 out of 4 would be considered affected, so that'd be 25%. So we've got 25% that are normal, or 1 out of 4, 50% that are carriers, and another 25% that are affected. And then the last question says, describe the symptoms of the genetic disorder that you modeled. Well, this is just basically restating what's on page 7-11 in your lab manual. But sickle cell anemia will make you have abnormally shaped red blood cells because the hemoglobin molecule that is the major component of the red blood cell is not normal either. And so they actually are this kind of, they call it a sickle shape because it's that curved shape like a sickle or a moon. And so they are shaped weird, they break off in the arteries and the capillaries, they can clog them, cause tissue damage, things like that. It's a very unpleasant disease to have. But what's interesting about it is the carriers of the disease, so that 50% if you've got heterozygous, but the carriers are actually very resistant to malaria. And so in countries where malaria is prevalent, you will see quite a good bit of sickle cell anemia because the heterozygous individuals that are carriers tend to thrive and do better and are more likely to have offspring. And so they tend to carry along that gene, which means that 1 out of 4 of their children potentially could be affected with the disease. But because they tend to be actually healthier than the, quote, normal individuals, it is a perpetual disease that we see pop up quite a bit.